Okay, I think in the interest of time, we're gonna get things started, and looks like we started recording. Fantastic. So it's my pleasure to introduce Laura Hughes for today's departmental seminar. I just want to start with a few of Laura's biographical details. So she did her bachelor's and master's degrees in chemistry at Northwestern, followed by a master's in chemistry at the University of Cambridge. Did a PhD at Stanford in chemistry, working on building lipid bilayers on solid supports as a model for cellular membranes. Then she branched out into sort of the data science aspect of her skills and talents, spent 3 years at USAID as a data scientist, using her talents in data visualization and analysis to understand the effectiveness of international aid in Africa. In 2017, we brought her back to biomedical research when she joined Scripps, first as a staff scientist and then as a senior staff scientist. So Laura has been involved in a number of projects. Many of them have been affiliated with Christian Andersen's U19 Center, focused on system biology and infectious diseases, specifically with the data management and bioinformatics core, which she now leads as the core PI. So today, Laura's going to tell us about Outbreak.info, which is unique, I think, because the initiative to create it and the execution to build it were almost entirely driven by Laura and Karthik Gangavarapu, who was a joint graduate student with Christian. So Christian and I really only understood what was going on when they said, look at what we built. And we said, wow, that's great, do more of that. And since then, the leadership has really been up to them. And it's just been fun to watch. So I'm as excited as you are to hear about the latest developments in Outbreak.info and also get Laura's perspective on what we can learn about data science in the context of infectious disease pandemics. My last duty is to let you know that you can use the raise hand function in Zoom to ask questions. That's where we're going to take questions from at the very end. And with that, I will turn it over to Laura.
So thank you, Andrew, for that kind introduction. So today I'm going to talk to you about this platform that Karthik and I developed along with collaborations across the Andersen, Sue, and Wu labs to really discover and explore COVID-19 data. And so I think one of the things that's become very clear in the past year and a half is the importance of having good, timely, and understandable data, especially to be able to track and understand the virus spreading across the world. And this is particularly true in this pandemic, which has seen an unprecedented amount of data, as well as misinformation being shared across the world. And so while I'm going to be focusing pretty heavily on COVID-19, I think the challenges that we face in terms of sharing, integrating, and visualizing data really transcend any pandemic. And so at the end, I'm going to talk about how we're thinking about extending the tools that we've developed and applying the lessons we learned to other problems. And so to begin with, I wanted to begin with thinking about the scale of COVID-19. I think, it's really easy to lose track of just how big it's been across the world as we get inundated with the minutiae of the virus. And so I want to talk about scale in 3 dimensions. The first being the scale in terms of the number of people that this has affected. So in terms of the WHO public health emergencies that have been declared by the WHO, COVID-19 has the distinction of being the most deadly, with over 4 million people currently have died from the virus. And to put it in context of other major epidemics and pandemics, COVID-19 is the most severe infectious disease crisis in the past 100 years, with over 190 million cases around the world. And in the US, which has been particularly hard hit, at least 1 in 10 Americans have been infected with the virus. And so I think the scale of the problem, is really motivating to figure out, how can we address it and how can we use data to learn something about the spread of this disease. But the second dimension of scale that I want to talk about is how quickly it spread across the world. So within about 4 months of the virus being identified, every single country in the world had a case of COVID-19. And then the last aspect of scale I want to highlight is the number of waves that we are still experiencing with COVID-19 that are still continuing to present challenges. And these waves lately are driven highly by the spread of variants of concern, which show increased transmissibility, virulence, and immune evasion. And so, as I'm sure you're aware, in the US right now, this is presenting a problem with the Delta variant, which is rapidly spreading across the US and starting to lead to the uptick in cases across the US. So this is a map from Outbreak Info. On the left, you'll see the data for the number of cases per capita in early June across the US. And at this point, transmission had started to plateau. And so the number of cases was starting to become level. And so in the past few weeks, driven by this new Delta variant, we've seen cases started to rise, especially across the sort of lower Midwest as the Delta variant is spreading across the country. And so I think the other sort of dimension of scale that we need to highlight is how much the research community has really responded to this crisis and really, for the first time, I think on a global scale, adopted open science to be able to try to share what they're learning about the virus. And so the scale of this is really impressive. So within Outbreak.Info's resource library, we've been tracking publications, data sets, clinical trials, and more that have been generated during the course of the pandemic. And so since about April of last year, on average, there are about 2,000 new distinct resources that have been generated every single week. And so this explosion of information presents a huge opportunity for us to be able to learn from what other researchers are doing. But it also is what the WHO has termed an infodemic, in that there's so much information, and how do we actually make sense of it? And so just to put this in context, within the first 2 years of the HIV epidemic, there are about 3,000 papers published within those 2 years. And now with COVID-19, we see more papers than that being published every 2 weeks. And so it's really a challenge to keep up with that. And this is true with the data front as well. And so one particular form of data which has been rapidly being produced is the number of SARS-CoV-2 virus sequences, which have been shared in an open data repository, GISAID, and what I'm going to be talking a lot about. And so, with all these virus sequences, it presents an opportunity to actually use genomic epidemiology to track the spread of and evolution of the variants and in near real time, with over 2.3 million sequences having been deposited in GISAID as of today. But that also presents a huge problem in terms of the technical challenges involved with being able to process all this data, update it in real time, and make sense of what is happening with regards to these variants. And so I think, the scale and the absolute, I guess, unification of how the research community has banded together is exciting, but it also highlights barriers that we have to actually use open science. So the first two challenges, which I've already mentioned, are the sheer volume of stuff that's being generated and the challenges being able to keep track of that, as well as the velocity which the data and the publications are changing. So we need to be able to address the volume and velocity of these challenges to actually use open data. The third challenge is really with a variety of information that we're learning about COVID-19. So in order to be able to really understand the virus, we need not only to be able to look at publications or even data sets, but also look at things like clinical trials, protocols, and software to get a holistic view of what we know about this virus. And within data, of course, there's a huge variety of heterogeneous data that is relevant to be able to understand the spread of SARS-CoV-2, including everything from genomics to epidemiology to immune function and structure, as well as the sort of social dynamics of how people are mixing together and the environment in which this is happening. The fourth challenge we have in actually using this open science is the veracity of data. So with data being generated very quickly, that presents a lot of opportunities, but the question is, how reliable is the data? And so one thing I want to particularly highlight is with the genomic data, within those 2.3 million sequences, in an ideal world, we would have uniform, unbiased sequencing across the world so that we could track and understand how these variants are changing across each country. But unfortunately, that's not the case. And so in some countries like the United Kingdom, they're actually sequencing about 10% of the cases, whereas in other places like India, they're sequencing less than 1% of the cases. And so it becomes a challenge to understand how reliable the data is and to be able to compare between locations. And then the last challenge is really the decentralization of information. So if I want to be able to find information about SARS-CoV-2 and data around it, it quickly becomes a sort of scavenger hunt across the web, going to lots of different locations which have data, but not in formats that can talk to each other. So everything from publications to technical briefings from public health institutions to more sort of standard ways to share data, such as open data repositories like Import, as well as individual lab projects which are publishing their own data. And so being able to, first of all, find all this information, but then actually have it to talk to each other is a huge challenge. And so what I think is abundantly clear is that to be able to take advantage of the wealth of knowledge that we are generating and really bridge the gap between information and insights to be able to track the spread and evolution of the virus and variants, we need to develop better tools and infrastructure that allow us to delve into all this heterogeneous real-time and large data. And so what our group has been focusing on very heavily is building tools that really enable analysis, visualization, and sharing. And so we are trying to tackle three specific problems. The first being, how do we discover data? So dealing with the volume, the velocity, and the decentralization. How do we actually find information that's relevant to study these variants? The second question is, how do we actually harvest, harmonize, and integrate data so that we can get them to talk together and to merge data across different sources? And the last is really, how do we put it all together to start to form hypotheses to understand what's happening with this pathogen? And so this obviously incorporates all the challenges with using open data. And so this is where I'm going to start sort of walking you through how we're approaching supporting hypothesis development. And then afterwards, I'm going to take a step back and talk about technically how we actually deal with harmonization and integration, and then data discovery writ large. And so to do this, as I mentioned, we've developed a tool, Outbreak Info, which really unites information about COVID-19 in a discoverable and explorable platform. And so there are three major components of data and tools that we've developed. And the first is a standardized research library. And so this unites almost 190,000 individual resources, including publications, clinical trial data, or clinical trial information, data sets, protocols, and more into one searchable platform so that you can search across the resources to understand the current state of research on COVID-19. The second aspect is an explorable interface of epidemiology data, which really allows you to dig into the case and death data at any geographic location, whether it's a country, a US state, counties, or metropolitan areas. And then the last is a project which is our most recent addition to look into real-time variant surveillance to do genomic epidemiology in near real time. So analyzing all the virus sequence data that we have and starting to integrate it with the epidemiology and research data to better understand these variants. And so I'm going to start here talking about how we've developed this tool to support hypothesis generation. And these are, of course, the wonderful Outbreak Info team who played heavily in its development. And so I think it's pretty clear right now that variants have altered the trajectory of the pandemic. And so these variants have been associated with rising cases across the world. And so one clear example of this is in the United Kingdom. And so you can see that there are currently 4 distinct waves. And the third one, the largest that they had seen, was driven largely by the prevalence of this variant B117, also known as the Alpha variant, which has been shown to be more transmissible. And so despite the fact that in the UK at this time, there were pretty heavy public health interventions, there was a huge explosion of cases driven by this more transmissible variant. And of course, more recently, we are seeing the same thing play out, but again, with the Delta variant, which again is more transmissible, leading to more cases across the UK. And the literature has demonstrated that one of the major reasons for this increase in cases is that these variants of concern have been associated with increased transmissibility, virulence, and immune evasion. And so, it's becoming increasingly clear that keeping a track on these variants of concern, both the ones that we already know and the ones that are emerging, is critical to be able to respond to and understand this pandemic. And so our goal in this was to develop an automated, near real-time, customizable variant tracker. And we've developed 3 tools that address 3 sort of major sets of questions. The first is very basic of what mutations are actually within a lineage and how do they compare with each other so that we can understand how variants of concern are related. The second is a location-based view, which allows you to discover within a particular location what lineages are circulating at any point of time to be able to understand what variants might be worth watching. And the third really digs into either a lineage or a mutation or a set of mutations to understand how is that prevalence changing and how is it distributed across the world. And so our goals in this are threefold. The first is to make sure that it's updated daily so that we can have the most up-to-date information that's possible to understand how these variants are changing. The second is to make sure that it's automated and customizable so that we can be able to delve into any particular location or any particular variant. And this is particularly important because we're not only interested in the variants that we already know about, but the variants that will continue to emerge over the course of the pandemic. And so we need to be able to have a way to be flexible enough to be able to track those as well. And then the last point is we want to make it visual and interactive to make it as easily interpretable as possible. And all of these 3 goals really drive to the overall goal of being able to support exploratory data analysis to really understand what's going on in these virus and variants. And so I'm going to break out now and show a quick demo of some of the features within Outbreak.info. And so I'm going to start with our location tracker. And so I'm going to start with a very basic question of what's been going on in California with these variants. And so you can develop a location report based off of any location and see what variants are within that location. And so the first view of this is a stream graph looking at what variants have actually been changing. And so I'm going to change this to look at the past 90 days, so the past 3 months of what's been going on the virus. And so if I zoom in on what's been going on from the fall to recently, if you remember in the spring, one of the things that was really concerning to us was seeing the explosion of cases of 2 related variants, B.1.429 and B.1.427, which are also known as the Epsilon Variant. And so at its peak in the spring, about 60% of the cases that we were sequencing in California were these 2 variants. But if you look at what's been happening more lately in the past sort of 3 months, collectively, these variants only compose about 6% of the cases that we've been sequencing, and even fewer in more recent data. And so the question is, what happened? You know, why did we see this these lineages sort of increase and expand and then be taken over? And so the first part of the story seems to be the prevalence of this alpha variant, the B.1.1.7, which is more transmissible. And so this has rapidly expanded in the spring and summer and became the dominant lineage in California. But that's not actually what's happening right now. And so right now what we're seeing is 2 related lineages which are starting to expand. The AY2 lineage and its parent lineage, B.1.6.1.7.2, which are collectively known as the Delta variants. And so this gives you a view into what lineages are expanding. The other parts of the variant report include a comparison table so that you can see which mutations are within the variants which are currently within that location, as well as a table giving the overall prevalence of variants of concern and variants of interest. And then a tracker which starts to look at the prevalence of these variants and start to relate it to epidemiology cases. And so we can actually sort of delve into the story that we saw before. So if we look at the expansion of B.1.429, you see it rapidly progressing in the spring and then tailing off in the summer as B.1.1.7 comes in. And then as the Delta variant comes in, we see a dramatic increase in the prevalence of B.6.1.7.2, the Delta variant. And that seems to possibly be related to what we're starting to see, some cases starting to increase within California. So this report allows you to really discover what's going on with the variants. And then the other 2 tools that I'm going to show allow you to delve into more of the details. And so the first thing I want to do is to be able to look at these 2 Delta variants, so the AY2 and the B.1.6.1.7.2, and ask a simple question of how are the Delta variants related? And so to do that, I'm going to go into the lineage comparison tool, and I'm actually going to change the lineages. So the default view is to look at all the variants of concern, but I'm going to remove the ones that are not related to Delta, and I'm going to add in AY3, which is another Delta variant. You can also change it to look at any particular gene that you want. So I'm going to focus in on the spike domain. And what you can see is a heat map looking at the prevalence of these mutations across the Delta variants. And what's very clear is in the purple, which shows almost 100% prevalence of these particular mutations, there are actually 6 mutations, including the P681R, which is a mutation of interest, and the L452R1, which is also a mutation of interest, which is shared across these lineages. And so one thing that I think really distinguishes Outbreak.info is that we can actually create a report which combines these lineages together by looking at the cross-section of these 6 mutations. So next, I'm going to create a customized mutation report, and where I select my own mutations. And so I'm going to look at these 6 mutations that are shared across the lineages and create a report to track their prevalence and where they're actually located across space. And so it takes a minute or two to retrieve the data from our API. The first thing you see is a breakdown of mutations within that I've selected and the lineages which they are prevalent in. So this combination of 6 lineages is prevalent in at least 70% of all the Delta lineages. There's an overview table which gives you a breakdown of the number of sequences that have been detected worldwide. All of these locations can be changed so that you can customize them. And so we can look at things like how the Delta lineages combined are progressing across the US. And what we see is that they have, of course, as we're seeing, are dramatically expanding, especially in the past few months. And then there's a geographic breakdown, which again highlights the fact that in sort of the lower Midwest, as well as the Mountain West, is where we're seeing the leading edge of the Delta variants starting to expand. And so this really gives a lot of tools to be able to track and understand the variants to support hypothesis generation. And so one of the things that you can start to do is ask questions of the data to look at, trying to develop these hypotheses. And so I'm going to break through 3 design principles that we have when we are designing Outbreak.info to focus on how we decided to display this data. And so the first is, again, trying to support exploratory data analysis. And so one of the questions that I had looking at the California data is that we clearly saw this sort of inversion between the alpha variant and the delta variant, where as delta was introduced, it quickly is starting to take over any B117 prevalence. So the question is, how generalizable is that? Is there actually evidence that B117 is more transmissible than the delta variants? And so we can pull data from across the world. So looking at the United Kingdom, we see a story where we see alpha dramatically increasing to the point of, in early April, about 99% of the sequences that were being generated were B117. And of course, in the past few months, this is almost completely flipped, where in early July, 99% of the cases that were being sequenced were delta and less than 1% were alpha. And we see this competition between these 2 lineages being consistently played across the world, where I'm showing the top 11 countries that have been sequencing the most. And in all of them, you see at different times this inversion of B117 shown in blue and B16172, the delta variants shown in orange, where delta seems to be outcompeting alpha in almost all cases. The one sort of case that seems to not fit this trend is India, and that's because at the point at which the delta was introduced, B117 prevalence wasn't that much, but still it seemed to outcompete all the other variants. And so the other important point about Outbreak.info is that, not only do we have genomic data, but we also can connect it to epidemiology and research to better understand these variants. And so within our research library, we can search for any publications or data sets which may talk about the alpha and delta variant. And so we can see that there are 37 results, which you can then explore to find publications like this from our collaborators at Helix, which are looking at the displacement of B117 by 6172, or this other paper looking at a meta-analysis of vaccines to understand how these variants of concern are actually affecting vaccine efficacy. And so all of the data within the research library is then linkable out to the actual publications or data sets so that you can actually delve into the data. And the last component of supporting exploratory data analysis and hypothesis generation is that all the data that we compile and collate and standardize is available for downstream analysis. So we've created an API which allows you to access the information. A talented undergrad, Manar, also created an R package, which allows you to both access the information as well as recapitulate some of the visualizations within Outbreak.info, as well as within the site itself. You can actually download all the data linked to the visualizations as well as the visualizations themselves. And so being able to actually look at the raw data and being able to play with it. And so the second philosophy that I wanted to highlight is we really wanted to make sure that our reports were both customizable and interactive. And so the importance of this is to be able to really delve into anything that may be of interest with these lineages. So currently there are 1,290 separate pango lineages and about 31,000 distinct mutations that occur in over 10 sequences within our data set. And so we want to be able to support any of the over 40 million combinations of lineages and mutations so that you can look at things like a P1 lineage report or P1 with the POO mutation or the P6181 mutation itself. And so we wanted to make it customizable to explore any combination of lineages and mutations. And then the second aspect of customizability is to be able to look at any of the 2,300 locations within that have been sequenced. And so this allows you to be able to dig into what's happening in a particular country or a state or province or even a US county to look at, in this example, how the Delta plus variants are proliferating over time. And so what I really want to emphasize here is that this allows you to start to make comparisons to understand how the spread is different between locations or also within one particular location. And we're seeing that right now with Delta, starting to have its spread in the Midwest, whereas other places are seeing that spread happen at a later date. And so the customizability allows you to really delve deep into any of these locations. And then the third thing that was very critical to us was to be able to try to convey uncertainty and biases within our data. Now, in an ideal world, sequencing would be unbiased, but unfortunately, that's not actually true. And so sequencing is not random, and it's not random in at least 3 ways. So the first is over time. Unfortunately, it takes time to actually sequence the data. And so more recent data is less reliable than historic data just because there are fewer samples. The second is between locations. So as I highlighted from the start, we see rapid differences with countries like the United Kingdom sequencing a lot and other places sequencing very little. And so comparing between those locations becomes challenging as places that aren't sequencing very much are not as reliable. And the third is within locations, there may be biases that determine how sequences are generated. And so if, for instance, cases of particular clusters of outbreaks or a particular type of case, like a breakthrough case from vaccination, are sequenced more frequently, then those are going to bias our results. And so our goal is really to try to balance being able to provide the information with also recognizing the fact that these measurements are inaccurate and uncertain. And so I'll be the first to admit that, we still have a long ways to go with representing uncertainty. And I think this is a really active aspect of research to try to figure out how we can actually convey the uncertainty. And so our approach right now is to, first of all, with any measurement that we make, we provide confidence intervals to provide bounds for the data and show where we think it's more or less reliable. So for instance, in this graph showing the AY2 prevalence within Nevada, it seems like overall the prevalence is hovering about 10%. But you can see by looking at the confidence intervals that this actually ranges from anywhere from about 2% up to 20%. And to highlight this even more, we show a histogram at the bottom of each graph showing the total number of samples sequenced in every day, which really highlights the fact that in more recent days, there hasn't been much sequencing going on in Nevada, and so that's why you see the confidence intervals blowing up. We repeat this process within the location reports. So looking at the stream graphs, comparing all the different lineages over time, you can again see that San Diego, unfortunately, hasn't been sequencing many cases lately. And then within any tables that we provide, if there are not a large number of samples being generated, we actually decide not to show any estimates, instead providing the raw numbers. So in this case, in Kansas, there's been one AY2 sequence which has been found, but we don't think that there's enough samples to be able to provide a good estimate of its prevalence within Kansas yet. And then lastly, within all the maps, we do a similar thing where if there haven't been at least 25 samples within that location, we actually gray out its cumulative prevalence so that it's not distorting the view of the map. And again, this can be changed so that if you want to, you can actually look at all the data or even set a higher threshold. But by default, we're sort of hiding the data that we think is less reliable. And so I think something that's been really exciting to us has been seeing, over the past few months, how Outbreak.info has been rapidly adopted by the community. So just to give you a few numbers in terms of scale, the last month we had over 39,000 individual users with 349,000 page views. And over 5 million different requests to our API, both from Outbreak.info as well as programmatic access through the API. And Outbreak.info is commonly cited in the media as a way to keep a handle on what's going on within a particular variant in a particular location, as well as being integrated with research tools. So things like covelineages.org, so these are the people that maintain the Pango lineage designations, actually link to Outbreak.info, as well as Cove Spectrum and GISAID. And then of course you can also find lots of interesting examples across Twitter about how people are using it, including this researcher who back in April was pointing out the fact that India unfortunately was seeing the rapid proliferation of the Delta variant, and our own Eric Topol, who's pointing out the increase of Delta within Japan, and thinking about how that perhaps plays into the Olympics coming up. And so to sort of wrap up this part, I wanted to sort of emphasize what I think the strengths of Outbreak.info are. And so the first is that it's a standardized resource which collects data from over 18 different locations and puts them in a form that they can start to talk to each other. And this encompasses everything from research, including publications and data sets, to the epi data, to the variant data. And then second, it's interactive and real time so that we can actually delve into deeply to understand what's going on in these variants and track how they're changing over time. So with that, I want to sort of talk a little bit about the actual logistics of how we actually go about this. And so I'm going to talk about our efforts to build a sustainable and extendable data pipeline and how we actually harvest and harmonize all these data. And so I think the importance of this is pretty apparent by the fact that SARS-CoV-2 continues to evolve, both the virus itself, as well as the data and the research that helps us understand it. So when we launched the variant reports in February, we were tracking 3 variants of concern, the alpha variant, the beta, and the gamma variant. And in the subsequent months, there have been a lot more variants of concern and variants of interest that have been added. And so we need to be able to adapt to changing data, to be able to incorporate both new sources of data as well as changes within the data. And so how we do that is we've built a pretty robust data pipeline that actually analyzes the data. And so one example of this is our Bjorn Pipeline, which calculates the mutation prevalence for every single lineage within GISAID. And so what's really remarkable about this is that it's able to process 2.3 million records every single day in less than 2 hours. And so how this works is that we actually suck up all the data from GISAID, we divide it into chunks of 10,000 sequences, and then we go through a process where we do quality control to filter it based off of sequences that are poor quality. We then do a multiple sequence alignment and some metadata standardization to, again, remove poor metadata that allows us to provide mutation counts of every lineage and every mutation within the GIS8 repository. This then gets filtered into our distribution pipeline, anchored by BioThings SDK and Outbreak Info. And I should mention that this is a collaboration with the Anderson Lab, with them really being responsible for developing Bjorn and making it able to process all this data within a short period of time. And so this feeds into our sort of larger data pipeline to distribute standardized heterogeneous data daily. And so the sort of ingestion and harmonization process that I showed for Bjorn is really replicated with our other sources of data, including the epidemiology data and the research library, so that all of these get merged together. And using BioThings SDK, which is a platform developed by Chun-Li Wu to be able to take data and quickly make an API out of them, we then harmonize and integrate all these data into one uniformed API, which then goes and serves both the front end and our R package. And so I think what I really want to emphasize about this is that because we've invested so much in the infrastructure to be able to process all these data, we're able to do this daily, and we're able to add it in new data sources very rapidly. And so the process of adding new data sources was actually done really incrementally. And so when we started back in March of 2020, we started with the epidemiology data, so looking at cases and deaths. And then over the subsequent months, we added separate parsers and integrators to actually integrate in all the research data library. And then back in February, we were able to add in the variant reports. And so because we had developed this robust and extendable data pipeline, it was actually fairly easy to incorporate the genomic sequencing. And so within a span of about 2 to 3 weeks, we were able to be able to integrate all these sequences into Outbreak Info. And so the last part of this sort of broad, robust, extendable system is really to focus back on how do we actually find information and work on data set discovery. And so I think the strength of this is really to be able to merge automated processes with human curation. And this work has been led by Jinder and Marco. And so the magic of how we are able to integrate data sources together is to really use rigorous schemas which allow our data sets to be integrated. And so we are actually basing this off of schema.org, which is a widely adopted standard used across the web to describe information. And so the advantage of using something like this is that if you have a data set which is described using the schema.org dataset schema, that means that it can be picked up by automated data crawlers like Google Dataset Search, so that we can allow all of our data sets to be found very easily. And so what we have actually done, Jinder in particular has been working on this, is building out these robust schemas which allow us to merge data from different sources. And so these schema really compose 3 parts. So the first is the fields that are required by Google Dataset Search to be able to be found within Google, but then also reusing community standard schema.org fields, and then incorporating fields which are relevant to infectious diseases so that we can capture those properties of data sets which are most pertinent to infectious diseases. And so I'll be the first to admit that I find schema both really obnoxious and really boring, which is always a bad combination, but they also are really powerful. And so the first thing is that because we have a common structure amongst all these data repositories, it means that we can have a unified way to search across data repositories rather than having to go to individual ones and find them. And the second is that it opens up the opportunity to allow web crawlers to be able to find and access this information. So not only do we get the benefit of having this nice standardized data, but we're actually opening it up to the world so that anyone else can find it and use it. And so it seems like a lifetime ago because this was before COVID, but we started with sort of a thought experiment of, can we actually use these schemas to actually integrate infectious disease metadata across different open data repositories? And so with Andrew Su and Chun-Li Wu, we developed 3 sort of sets of processes, going from as automated as possible to hand curation that allow us to integrate data together. And so the first is writing crawlers to pull structured and exposed data from places that already use the schema.org standard like OmicsDI and Dataverse. And then also incorporating other open data repositories which have structured metadata, but not in a schema.org format. And then also adding in human curated ones from individual projects. And so we developed all of these together to combine them into one searchable data portal and also accessible by Google Dataset Search. And so within this project, which we called the NIA data portal, we were able to import and access over 650,000 infectious disease data sets. And I think the point of this is really to have a prototype for a discovery platform, almost like a Google for infectious disease data, so that you can start to be able to find all this information scattered across the web. And so this was really the inspiration for the Outbreak.info research library, and something that I think we want to continue to develop to be able to increase the availability and utility of open data that already exists. And so with that, I want to end with discussing what I think is next with sharing, integrating, and visualizing data. And so to start with, I want to talk about immediate plans for what we're planning to do with Outbreak.info. And so I think the real focus over the next few months is working on data integration and analysis to move beyond just looking at sort of in a bioinformatics way what's happening with the variants, but actually starting to think about how this is functionally relevant. And so the importance of this is not only to track the variants of concern that we already know about, but also to track the emerging variants, which we know are going to continue to arise. And so there are sort of 5 categories of things that we want to continue to do. So the first is to be able to develop complex mutation and lineage queries to better understand the variant growth dynamics. And so this is things like automatically finding the mutations and homage between a parent and its sublineage so that you can do things like track all the Delta variants at the same time. We also want to incorporate phylogenetic information to understand viral transmission and evolution, and then start to add in structure, functional, and immunological data to really understand what the functional implications of these mutations within the variants are, and to try to tease out why particular variants are growing. And so using all 4 of these things, we want to also be able to compute variant growth rates so that we can develop an automated, customizable way to develop a watch list for variants which seem to be emerging. So I want to emphasize too that we want these to be customizable in 2 dimensions. The first, to be able to develop watch lists for any particular location, but also opening up the parameters that we have filtered all of the 1,290 different lineages so that users can customize what they think is most important in terms of variant growth, whether it's the raw growth rates or a particular structural or functional change within that mutation. And then lastly, we want to integrate more data that gives us a more holistic view of understanding how the virus and the variants are changing. So obviously, within the spread of the virus, there are many contributing factors as to whether a particular variant spreads. And so we want to start to understand the complex interaction between the social dynamics and policy, the public policies that are in place, along with the biological immunity and virus genetics. But I think more broadly than that, we want to expand and think, how can we use this experience to start to build infrastructure that allows us to tackle the next problem, whether it's a new virus outbreak or a new emerging virus that evolves, or more broadly, how do we integrate, share, and visualize data within bioinformatics writ large? And so to go back to this, I want to go back to the 3 sort of themes of areas that we wanted to improve with our tools. So focusing on dataset discovery, integration and harmonization, and hypothesis generation. And so thinking about what we've learned and continuing challenges that we have. And so I think what's become very clear in terms of promoting dataset discovery is the importance of making it easy to share data and to be able to develop automatic processes to harvest where you can, and then supplementing with human curation where you can't. And the thing that I really want to emphasize is that I think in order to encourage people to share data, you really need to give them an incentive to do that. And in my mind, that all comes back to developing tools that actually add value to the data. So one of the best use cases that I see of this is really what we've seen with the GISAID virus sequencing data. So of course, a virus sequence in itself has intrinsic value, but its value becomes much larger when you actually are able to combine it with other virus sequences. And so I'm hopeful that tools like Outbreak.info encourage people to actually deposit their data in public repositories by allowing them to use it and see how it integrates with other data. In terms of harvesting, harmonization, and integration, I think it's very clear that we need to be able to combine to common standards so that we can actually crosstalk between data sources. And then lastly, in terms of hypothesis generation, I think it's abundantly clear that we're only going to continue to have an increasing amount of data with increasing complexity and from various different heterogeneous types. And so in order to be able to understand how this play together, in my mind, that's really exploiting the tools of data visualization and human-computer interaction research to help us, in the lovely words of John Tukey, help us be able to notice what we never expected to see. But with that being said, there's still a number of challenges in these three areas. And so the first with data discovery is really, how do we do it sustainably? So how can we do it at scale every day, combining from different sources? In terms of integration, I think it's one thing to be able to merge a lot of data sets together. The next step beyond that is, how do we actually connect data set discovery into analysis? And how do we truly support integrated systems biology analyses that take in multiple forms of data and actually interpret them meaningfully? And the same challenge, I think, exists with hypothesis generation of how do we represent integrated multidimensional data. I think both visualization and analysis have developed tools that are particularly good at either analyzing or representing one type of data, but to actually combine different types of data, I think, is still an active area of research that we need to continue to develop. And then lastly, as I talked about, how do we actually convey uncertainty within our measurements to be able to provide a realistic but still interesting view of what's going on with the data? And so I think our vision for how we want to continue to expand the Outbreak.info platform is to really develop it into a robust discovery and exploration platform that acts as a bridge to analysis and modeling. I think it's very easy to get excited about machine learning and AI tools to be able to analyze data, but in order to be able to actually exploit those tools, you need to have good data that's findable in a format that can be integrated so that you can do those complex interactions. And so I think we're thinking about how to expand our platform to find the data, to explore it, and then lead to the analysis that will hopefully lead to interesting insights about the data. And in my mind, the common thread that sort of integrates all of these challenges together is to be able to use visualization as a tool that helps us analyze, integrate, and share data. And so visualization, of course, lies at the heart of science. Everything from, John Snow plotting cholera cases in Soho in the 1850s, to Ramon Cajal drawing beautiful diagrams of neural networks by hand, of course, to Franklin, Watson, and Crick looking at, the nucleotide structure in 3 dimensions of DNA. And so despite its importance, though, within science, I think that scientific visualization research really remains underdeveloped. Visualization research, I think, tends to focus on other domains rather than being applied to science. And so the consequence of that is that the lessons learned from visualization, design, and UX principles are inconsistently applied in scientific data to explore and explain data. And of course, 2 particular challenges that I think exist within this diverse, heterogeneous, complex data are being able to combine different data types together and represent uncertainty. And so I think the solutions to all of these challenges we present really sit at the interface of domains. And so I'm really excited to continue to look at the intersection between data science and technology, as well as visualization and design, to answer these really tricky questions we have in science that allow us to enable modeling or to inform public health interventions, understand disease mechanisms, and evaluate treatments and vaccines. And so the point I want to leave you with is that having good structured data and tools really allow us to use data to solve these tricky problems. I think over the past year and a half, it's obviously been challenging for a lot of the world in a lot of different ways. But something that has consistently given me hope has been seeing the scientific community respond to these challenges and really work together to try to address them. So, having researchers work all hours through the night to, collect samples, to analyze the data, and to share them more broadly. And so something that really has been coming back to me is this passage from the Emperor of All Maladies, which is a bit of a strange connection, but it's been on my mind as I've been thinking about, the rapid proliferation of open science within bioinformatics and with COVID-19 in particular. And so if you haven't read The Emperor of All Maladies, you should, because it's a fantastic book and there's a reason it won the Pulitzer Prize. But in it, Mukherjee makes this comparison between the war on cancer in the 1970s, which unfortunately was not as successful as anyone would have liked, and these other big sort of scientific endeavors like the Manhattan Project or the Apollo program. And the distinction he generates is that within those that were successful, like the Apollo program, all the pieces were already in place. So there was a solid foundation of knowledge. And what was needed was the mobilization of money and people and facilities to really connect all the dots together to put together in one imposing package the scientific knowledge we already possess. And I think this analogy is really true with what we're seeing in bioinformatics right now. I think we have a situation which is primed to be able to make connections between pieces of data that already exist. So we have the ability to do cross-disciplinary experiments. We can generate lots of data. We can run the analyses. And I think most importantly, we have the will of the community to be able to share them and think about analyzing them together. And so really the missing piece is to be able to continue to develop tools that allow us to better share, integrate, and analyze these data. So I want to end, and I want to leave plenty of time for questions, but of course I want to thank the entire Outbreak.info team. If you know any of them, they are a very diverse group of scientists and engineers who really unite their unique talents to be able to develop, I think, I'm of course biased, but what I think is a pretty amazing resource to be able to explore variants. And I particularly want to highlight the contributions of Karthik Gangavarpu, who really sort of led this project with me as we sort of had this idea back in March of, can we actually start to build a data platform to explore what's going on in COVID? And so lastly, I want to thank everyone in the Sue, Wu, and Anderson labs, as well as our funding agencies, particularly NIAID, as well as the data providers who really make all of this possible. So with that, I will stop there and open it up for questions.
Great, fantastic, Laura. Thank you so much for that seminar. Okay, so reminder, please raise your hand or click the raise hand button if you'd like to ask a question. Alternatively, you can type questions in the Q&A box, and maybe we'll start there as people raise hands. Jordan Willis asks if there's any chance for a Python package in tandem with the R package.
It's in the works.
In the works. Fantastic.
Yeah, and so I will say that so Chunli Wu, who is our collaborator, has developed an easy way to access information from BioThings API, and so our plan is to extend that to provide a Python package to be able to access all the information.
Fantastic. Next, we have a question from Jamie.
Hi, Laura. Thanks for the deep dive. That was
quick.
I mean, it was really interesting. But here I'm kind of finding myself horrified because I'm an experimentalist.
Yeah.
And basically what you've done is just given all your data away. And whereas we tend to have very small amounts of data that we husband very carefully and keep from our competitors. Mm-hmm. And so I mean, this is a real different paradigm that's pretty impressive. And so I'll just ask a general question. How do you get us to do and think the way you're thinking?
Yeah, I mean, I come from a similar background to you. In chemistry, sharing data and even sharing code was not the norm. And so I think to really be able to start to shift this paradigm, I think you have to provide incentives for people to do it and reasons for it. And to me, that comes with seeing the possibilities to be able to do cross-disciplinary analyses that integrate lots of data together. And so, it would not be possible to track the variance without having the contributions of so many different labs. There are over, I think, 1,000 different contributors that are actually contributing data to GISAID. And so this is something that we can't actually do alone. And so that's where I think the analogy to the Apollo program is really apt, because in order to tackle these really complicated questions, we need these multidisciplinary approaches that incorporate lots of different data of different types and different areas of expertise into one. And so that's where I've been really lucky to be able to work in groups like CVISB, the Systems Biology Consortium that Christian Andersen put together, that really, I think, unites talents. And so I think the philosophy that he and Andrew and others have is, why compete when you can collaborate? And so I think having that become the norm and having people see the benefits of being able to not have to reinvent the wheel and go through the process of developing all these code yourself, or doing all these hard experiments is the path forward. But I agree, it's an evolving process, and I think that's where COVID-19 has been really interesting to see so many people across the community really start to adapt to this paradigm.
Thank you.
Fantastic. Joel, let me see. Joel, do you want to ask your question?
Yeah, the biggest problem in my perception, and I think in healthcare, is not generating the data, but is getting the data from people like us to people who actually have to use it. Mm-hmm. And you describe a system that requires a certain degree of sophistication in accessing the data in the formats in which you provide it. Now, the assumption may be that the current generation of physicians who are being trained will be thoroughly facile in these approaches, so it won't be a big problem. But even if they are, the next problem is, how do they have time, and how do you envision getting around the time limitation of confronting a problem and being able to access the data? I mean, certainly in my own experience, ultimately accessing the data before I can even think about it is something of a barrier, and certainly much of uses a lot of time.
Yeah, I think at its core, that's really the challenge that we're trying to address, is how do we decrease that barrier so it's easier to use data? And so I think the two things that we are trying to do is, first of all, to develop standards and sort of do the hard work to merge all this data together so that you can start with a clean data set rather than having to go to 18 different sources and combine them yourself. And so again, that still implies that you're the type of person that wants to sit down with Python or R and delve into the data. And so I think that's one aspect of decreasing the barrier. But the other aspect, I think, is really to start to develop visualization tools which allow you to explore the data without having to touch any part of code. And so I think that's where Outbreak.info allows you, without having to actually deal with the data itself, explore trends over time, see how variants are changing, and see how they're distributed geographically. And so I think our approach is really to try to be as flexible as possible, to spend all the time, dealing with standardization, but then opening it up and providing tools that allow people to access it, whether it's the raw data or an interpretable interactive report that allows you to view the data.
Great. Libby Getzoff, do you want to chime in with your question?
What an awesome talk. It emphasized all the things that I think are really great about scientific research, integrating and collaborating and visualizing and outreach. So my question is, the things that you showed with visualization were two-dimensional. Are you dealing with three-dimensional or more dimensional aspects of visualization as well?
Yeah, that's a great question. I think that, to some extent, you need to delve into multi-dimensions because, we don't deal with just 2 dimensions of data. We deal with multiple aspects. But that being said, there's been lots of research going back to the 1980s that showed that humans are not really great at processing information in 3 dimensions. And so I think the question becomes, how do we develop tools that allow us to explore multiple dimensions without having to resort to 3-dimensional objects that people are bad at interpreting? And so in that mind, in my mind, it really comes down to looking at things like dimensionality reduction, so analysis techniques that allow you to incorporate multiple dimensions of data, but also using visualization techniques that allow you to explore visualization in multiple levels. And so using techniques pioneered by Ben Schneiderman from Maryland to be able to see a high-level view of the data and then delve into the details. So I think in the scientific community, we think a lot about visualization in terms of like a Nature paper and a 9-panel figure with like figures that are, very small that you have to zoom in 300% to be able to view. And I think that that's really limiting in terms of what we're doing. I think that by embracing interactivity and allowing people to step through the multiple dimensions of data sequentially, rather than trying to have to look at all the dimensions at once, I think is really the next forefront of scientific visualization.
I agree with everything you said, except as a crystallographer, I would argue that data is a good
okay. I apologize for that. I mean, of course, 3-dimensional interactions are essential, but especially my background is in chemistry, and so I think a lot about how do things interact with each other, and of course that interaction happens in space. But that being,
condensing dimensions so people understand them is also a wonderful goal.
Fantastic. Great discussion. In the interest of time, I think we should cut it off there. I'm sure Laura would be happy to answer any other questions via email or at hopefully the next in-person event.
Yeah,
fantastic. Thank you all.
Yeah, I was going to say, I love talking about data and visualization, and I could stay here for hours, so feel free to email me at any time.
Fantastic. Excellent. Thank you, Laura, for a fantastic seminar. Thank you, everyone else, for the discussion, and until next time. Thank you.